I'll be answering question 1 for the final assignment, which is curing malaria through rational enzyme design. So first, we're going to talk a little bit about the protein of interest, which is human peroxoreducin 2, or PRX2, to be that's its abbreviation. It is an oxidoreductase enzyme, which means it performs a redox reaction in the cell. It belongs to the widely present cysteine-dependent protein family, which is a peroxoredoxin, and it's only the second isoform in this family. There's plenty of isoforms. I think there's 7 or 8, but this is the second one. The diagram on the right shows its catalytic and regulatory functions. It is specifically, we're looking at this inactivation function here, where it reduces a peroxide to a hydroxide, and we're looking specifically at hydrogen peroxide to water reduction. So why does it do this? The main reason it does is because hydrogen peroxide is a reactive oxygen species, which disrupts the redox or ionic balance in the cell. And this can have detrimental effects on oxidative phosphorylation and other vital cellular processes. It is peroxyredoxin 2's job to reduce this ROS, this reactive oxygen species, to restore this redox balance. It has also been seen that in the in excess of H2O2, there is an inactivation of PRX2's oxidoreductase function due to hyperoxidation. And we'll talk a little bit more about that in the future. So this is the first isoform of PRX, which is just PRX1. It is very similar structurally to PRX2 with I ran an embossed stretcher analysis, and it showed an 89.4% similarity with about 76 to 77% identity, which is pretty high for it. Although other papers did say it had a 97% similarity, which might be because this is a different version of the enzyme, or it might be bound to a substrate, which I'm unaware of. But despite the similarity, it is at least 10 times as resistant to hyperoxidation by the peroxides than is isoform 2. And Plasmodium falciparum, which is the parasite for malaria, imports these from the host's blood and incorporates it into its own cells. And because it is highly resistant to hyperoxidation by the peroxides, it is unable to be affected by the immune attacks by the host when due to it is unable to be affected by the immune response, which is led by peroxides. So that's one of the drawbacks of being resistant to H2O2 mediated hyperoxidation. And it would be in our best interest to design a mutant of PRX1 that is more susceptible to inactivation by these peroxides. So how do we go about designing a non-resistant mutant? Our first step would be to align both PRX1 and 2 to compare where they differ structurally. Because PRX2 is not resistant to H2O2 hyperoxidation, we should try to find the residues of PRX1 that have been altered, and give it its resistive properties. So yeah, one strategy might be to make PRX1 more similar to PRX2 by replacing residues. By replacing, I mean by mutating residues and drastically alter it. This could be ideal because only 10% of the residues actually that actually differ, and therefore even a fewer percentage actually contributes to this resistance. So it's not that difficult to actually um, to actually figure out which mutations can cause it um, to regain that sensitivity to the peroxides. And I've noted a few potential mutations uh, that I got from this image here. This image is from Emboss Stretcher, and it shows where where there's differences in the peptide structure, in the polypeptide structure of PRX1 compared to PRX2. It is especially necessary to preserve the cysteine at position 52 because that is an essential contributor to the peroxidase functionality of the enzyme. So yeah, these were some of the mutations. I think some of the more some of the more interesting ones were the lysine to glutamic acid substitution at 168. Um, yeah, because you're going from a basic residue to an acidic one, and so that's probably gonna play a big role. But there was also a not protein, proline substitution to valine, and while there may not be a very high difference in pKa of the side groups, proline has a very it has an odd structure. It's cyclic, but the amino group is not primary. It's a secondary amine, and therefore it can cause disruptions to the secondary structures such as alpha helices or beta sheets. But when it's substituted, it may we may be able to recover these structures. So let's see what happens. How do we test the inactivation of this mutated PRX1? Although because that's our main goal, right? We want to see if our mutations were able to sensitize PRX1 to the peroxides. We could use an in vitro model for this study. So cells could be cultured with the mutated PRX1 and wild type cells for comparison, which would include the non-mutated version of the gene. Hyperoxidation assay, which I got from a paper, Bolduc et al. 2018, um, can be used to determine the C hype 1%, which, uh, to be brief, is the concentration of peroxides at which 1% of the PRX is inactivated per catalytic cycle. So the higher this number, the so when this value is higher, it means it took a lot of the peroxide to inactivate a little bit of the the gene. So yeah, the reaction includes PRX um, at these stipulated concentrations, TRX 5 to 10 micromolar, TRX R 0.2 to 0.5 micromolar, and so on. The peroxide is added last because um, it's really potent, I guess. It works quickly. So this assay is performed for the wild type PRX2, the wild type PRX1, as well as the mutant PRX1, because we're comparing the activity of PRX2 to PRX1, but we also want to see how the mutated version of the PRX1 fares to either of them. And what we expect is that the PRX1 mutated should be similar or comparable to the wild type PRX2, and the PRX1 should be a lot more resistant, which means even a high concentration of peroxides will not affect the inactivation of PRX1. And so, yeah, we expect this C hype value to be the greatest for PRX1's wild type, but lower, a lot lower for the wild type of PRX2 and PRX1. Yeah, that's about it. Thank you for listening.